Hello everyone, welcome back to my channel. In this video, we are diving into the most exciting science and technology updates from June 23rd to June 30th, 2025. From space breakthrough to tech innovation, we have got the highlights that matter. Let's get started. First news is about Flavipiravir shows promise against Chandipura virus. Recent study have brought into light the potential of Flavipiravir as a treatment for the Chandipura virus, CHP. It was conducted by National Institute of Virology in Pune. These preclinical trials indicate that Flavipiravir can reduce viral load and improve survival rates in infected animals. So this flavipiravir can reduce viral infection and improve rate of survivals in infected animals, especially in animals. It is endemic. It means it is happened in particular area itself only. So it is endemic to central India and primarily affects children, leading to severe symptoms such as high fever and seizures. So it affects primarily affects children, especially children, leading to few symptoms are a high fever and seizures. About Chandipura virus, it belongs to Rhabdoviridae family and is transmitted by sandflies. So it is transmitted by sandflies and it was first identified in 1965 in Nagpur, Maharashtra. So this virus causes encephalitis. The main problem that this virus is giving is encephalitis. Encephalitis, the infection may be bacterial or viral. Inflammation, encephalitis means inflammation of the brain, which often due to the infection. So the infection may be bacterial or viral. In some cases, encephalitis may be the result of an immune system disorder. So leading to inflammation of brain, symptoms include fever, vomiting, altered consciousness and convulsions. About Chandipura virus, it belongs to the Rhabdoviridae family and transmitted by sandflies. So antiviral drug flavipiravir has been found. It has the potential to provide substantial protection against this virus, and the name of the virus is Chandipura. In the preclinical studies, which is conducted by Pune-based National Institute of Virology, means study of virus. So about transmission and vector, CHPV is primarily transmitted by female sandflies, and the name of that sandfly is phlebotomine sandfly, which are most active during the early monsoon season. So different types of flies are most active during monsoon season because accumulation of water here and there. So CHPV is primarily transmitted by female phlebotomine sandflies. Phlebotomine sandflies, which are most active during the season. The season is monsoon. Studies have indicated that certain mosquito species, particularly Aedes aegypti, could also transmit the virus. So first one is a sandfly. The name of the sandfly is phlebotomine, and second one is the carrier of this disease is mosquito, and the name of the mosquito is Aedes aegypti. It could also transmit the virus, CHPV virus, under laboratory condition. Under the condition, it is under laboratory condition. But however, no direct isolation from mosquitoes have been confirmed. So it is under, they have, researchers have found that it is under the laboratory only, not in the present scenario. Clinical manifestation of CHPV is its infection typically includes a rapid onset of fever followed by neurological symptoms, brain's problem symptoms, rapid onset of fever, and children under 15 years old are particularly affected. So the 2003 outbreak happened in Andhra Pradesh. In 2003, there was an outbreak in Andhra Pradesh brought into light the virus is having high fertility rates, high death rate among this demographic, with many deaths occurring within 48 hours after this infection. This CHPV virus, within 48 hours of hospital admission, there may be a chances of death. Treatment option, currently there is no specific antiviral treatment. There is no, because virus, there is a no treatment for a virus till now, because it is a puzzle for scientists, even though whether it is a living or non-living. So antiviral treatment for CHPV is, there is no specific treatment. Symptomatic management is a primary approach. What is the symptom the patient is getting? Focusing on maintaining airway, breathing and circulation. Fluid, to maintain fluid balance, the temperature control and seizure management are important to be provided to this type of patient. Favipiravir is being evaluated as a potential medicine, but clinical trials in humans are still pending. So clinical, it is happening in animals, but clinical trials in humans are still pending. It is going on. Process is still going on. There are few preventive measures. Preventive measures, CHPV infection involves vector control strategies. We have to control this vector. So this includes identifying and eliminating sandfly breeding sites. So we have to eliminate the area where the sandfly is breeding, growing. With the help of insecticides, we have to destroy that sandfly insects and eventually promote sanitation. It is required. So personal protective measures such as wearing long clothing and using insect repellents are also recommended to reduce the risk of sandfly. There is future research is going on. Ongoing research aims to develop a vaccine against CHPV. The National Institute of Virology is working on this, as current measures are insufficient to combat the high fertility rates, to mitigate the high fertility rates, to reduce the high fertility rates, death rates, especially among children. So efficacy of favipiravir will be further evaluated in animal studies before human trials commence. Second news about rapid detection of Nipah virus. Nipah virus poses public health threat due to its high mortality rates, high fertility rates, and potential for outbreaks. Recently, India made a major advancement in combating this virus, Nipah virus. The National Institute of Virology in Pune developed a portable test kit. They develop NIV in Pune develop a portable test kit that can detect the Nipah virus within few minutes. This innovation is important for high-risk areas like Kerala and West Bengal, where outbreaks of this Nipah virus have been frequent. Nipah virus is a zoonotic virus. It is primarily transmitted from fruit bats to human. And it can also spread through contaminated food or direct human contact. So the virus causes severe illness, ranging from there is mild respiratory symptoms to fatal encephalitis. The mortality rate can exceed 50%, making it one of the most lethal viral diseases, fatal viral diseases, which causes death. The newly developed LAMP-based point-of-care test kit, the name of the test kit is LAMP. It gives rapid results without needing a laboratory. So this kit is expected to enhance early detection, to detect it early, and contamination efforts in outbreak prone regions. and it is effective in the outbreak prone regions. So it has been patented and will soon be deployed in high risk states, soon be provided in high risk states. So this is about the Nipah virus, immune subject, how it is affecting, and its last, the recognition of non-overlapping epitomes, blockage of receptor, therapeutic protection in ferals. So historical context of Nipah outbreaks. The first case of Nipah virus in India was first observed and recorded in West Bengal in 2001. Subsequent outbreaks was reported in 2007 and 2018 with Kerala experiences with Kerala people facing multiple incidents. The virus has a history of causing fatality, causing death, with a 100% death rate reported during 2007 outbreak. Transmission dynamics. Nipah virus transmission occur through direct contact with infected animals, highly contagious, highly communicable, and also with contaminated food. In Malaysia, the initial outbreak involved pig farmers. In India and Bangladesh, human-to-human transmission has been observed, has been documented, particularly among caregivers and family members who were taking care of this type of patients. Symptoms of Nipah virus infection range from asymptomatic to severe neurological issues. Initial signs include the patient is having fever, headache, respiratory problems. So to diagnose, it's very challenging and due to non-specific early symptoms because it is asymptomatic at the early stage. Laboratory testing including RT-PCR and ELISA is essential for accurate diagnosis. So two type of test is available to know about the Nipah virus. First is RT-PCR and second one is ELISA. So currently there are no specific antiviral treatment or vaccine for Nipah virus. Supportive care is recommended for severe cases. So the Nipah virus is collaborating with pharmaceutical companies to develop monoclonal antibodies and a homemade indigenous vaccine for Nipah virus and to develop monoclonal antibodies against this Nipah virus. So the research aims to enhance treatment option and improve public health responses to potential outbreaks. Third news is about the Veracity Rubin's observatory impact on asteroid discovery. So Veracity Rubin Observatory in Chile has made strides in asteroid discovery. Before its official operation, it has already identified over 2,100 new asteroids. This powerful telescope is designed to repeatedly scan the night sky. To repeatedly scan this powerful telescope has the capability to repeatedly scan the night sky. It is expected to uncover millions of unknown celestial objects. It has the capabilities to protect Earth from potential asteroid collision as well. So functionality of the Veracity Rubin Observatory is to monitor the sky continuously, especially the night sky. Its large aperture and wide field of view allow for extensive coverage. This enables the detection of transient objects like asteroids and comets. The telescope ability to track moving objects is important to identify potential threats to Earth. So there is the importance of asteroid monitoring. It can pose risk if they come too close to Earth. There may be risk. So many asteroids resides in the Mars-Jupiter belts. Many asteroids reside in the present in the Mars-Jupiter belt. However, some have drifted closer over time. Some have drifted closer over time. The observation can track these objects and providing important crucial data for planetary defense. So understanding this trajectory helps scientists to assess collision risk, which is very significant. So the observation may also detect interstellar objects. These are space rocks. Interstellar objects are space rocks that originates, that develop outside our solar system and enhance our understanding of cosmic materials. Also give us potential to find such objects, which adds to the scientific significance of the observatory. About search for planet 9, the telescope may help locate planet 9, a hypothesized planet in our solar system. This planet is thought to exist far beyond Neptune. This planet 9, is thought to exist very far beyond Neptune. So its discovery would challenge existing model of our solar system as well. So finding planet 9 could provide important information about the evolution of planetary system, formation of planetary system. So there is a legacy of Verasi Rubin. Verasi Rubin was a pioneering astronomer who made contribution, who made important contribution, vital contribution to astrophysics. She was instrumental in the discovery of dark matter. So Verasi Rubin was instrumental observer in the discovery of dark matter. So his work demonstrated that stars at the edge of galaxy move faster than expected. This indicated the presence of unseen mass. So Rubin faced in these challenges also in a male-dominated field because she is a female, becoming a role model for women in science, especially in astrophysics. So observatories named in her honor, recognizing her contribution and resilience. So Vera C. Rubin Observatory is set to transform our understanding of, to understand our understanding of space. Its ongoing discoveries will update existing knowledge about our solar system. The potential finding could influence future research in astronomy and planetary science. Fourth news is about EMM negative blood group system. The International Society of Blood Transfusion officially recognized a new blood group system, and that is EMM negative, as well as it is also known as GUADA negative. So this designation emerged from the unique case of a woman from Guadeloupe, who is the only known individual to have this rare blood type. So the discovery put stress on advancement in blood transfusion, in transfusion medicine and genetic research. So what is GUADA negative? GUADA negative refers to the EMM negative blood group system, which is identified by the absence of EMM antigen on red blood cell. Because of absence of EMM antigen on red blood cell. So this antigen is a high incidence antigen and present in almost all humans. The lack of such a common antigen is medically and has led to the classification of this blood type as 48 globally recognized system in transfusion science. So the identification of EMM negative blood type began in 2011 when the women underwent routine, one woman underwent routine blood test. And initial attempts to classify her blood were inconclusive due to lack of technology. That time it is inconclusive due to lack of technology. In 2019, advancement in high throughput DNA sequencing, which allowed researchers to analyze her blood more effectively, efficiently. The unique genetic mutation responsible for the absence of EMM antigen was then confirmed. Following extensive validation, the ISBT recognized the discovery in 2025. So currently the women from Guadeloupe is a sole known carrier of the Guada negative blood. So she inherited the rare gene variant from both parents, not from a single parent. She inherited this rare gene variant from both parents. And this dual inheritance result in a complete lack of EMM antigens. So clinically, this means she can only receive blood from herself, as any transfusion from a donor with the EMM antigen could trigger severe immune reaction. So any transfusion from a donor with the EMM antigen could trigger severe immune reaction. So the discovery of Guada negative blood type has important implication for transfusion medicine. Individual lacking common antigen can develop allo antibodies, which possesses risk during transfusion. Recognizing such rare blood type is important for safe transfusion and help blood banks in managing rare blood group unit also. So this discovery marks the need for international rare blood donor registries, which can provide essential support in emergencies. Scientists are now focused on identifying others who may share the same genetic variant as the woman from Guadalajara. So this could enhance support for patients with similar rare blood types. Next news about newborn screening for sickle cell disease in India. So in recent years, India has made strides in addressing sickle cell disease through screening techniques. So a study conducted by the Indian Council of Medical Research, ICMR, brought into effectiveness of early diagnosis and treatment. This study was conducted by Indian Council of Medical Research. There is a major increase in sickle cell diseases in India. So this research spanning from 2019 to 2024 tested over 63,000 newborns because it is happening in the women who is pregnant. So tested over 63,000 newborns across high prevalent areas. So the findings suggest that early intervention can drastically reduce death rates associated with the newborn as is having SCD. So it can reduce death rates of newborn. So newborn screening for SCD is important. SCD is a serious inherited blood disorder which can lead to severe health complications, anemia, strokes, different types of infection, multi-organism affect, different types of organisms affected. So the ICMR study involved 7 centers in India focusing on tribal and high-risk region. So it aimed to identify cases of SCD and assesses the effectiveness of early intervention. The study found that 11.4% of tested newborns were carriers of sickle cell gene, while 0.9% were diagnosed with sickle cell disease. So benefits of early diagnosis provides many benefits. It helps for immediate medical intervention such as preventive antibiotics should be provided, regular health checkups and essential vaccination. Parents are educated on recognizing warning signals and symptoms, enabling quicker response to potential health issues. So, newborn diagnosed with SCD, sickle cell disease, receive comprehensive utmost care. This include the antibiotic is penicillin, prophylaxis, folic acid supplementation because of anemia. The newborn is suffering from anemia, so folic acid supplement is given, and hydroxyurea therapy. So such intervention, such care have reduced death rates from 20 to 30% to less than 5% in affected infants. It reduces mortality rates. Role of genetic counseling. Genetic counseling is an important process of a screening process because families must be informed about the implication of SCD and preventing measure should be taken for future pregnancies. So it is important in reducing and the incidence of STD in future generation. The study also aimed to understand regional differences in STD prevalence and the role of genetic modifiers. So it is essential for knowing the STD and why it is expanding these services across, how to expand these services across India. Newborn screening is important for expanding these services across India. So the success of this newborn screening initiative puts stress on the need for continued efforts in public health. By detecting STD early, India can improve health outcomes for affected families. So increased awareness and access to healthcare services are important in combating this inherited disorder. Sixth news is about snowflake yeast reveals into multicellularity. So recent study on snowflake yeast which have revealed intriguing vital information about the evolution of this organism. So this yeast which exhibits, which shows unique growth patterns, which is an important focal point for researchers to study the life form transition from unicellular to multicellular structures. So the findings suggest that physical processes may play important role in evolutionary leap, challenge genetic theories as well. So about snowflake yeast, it is a variant of regular yeast, grow in cluster rather than as a single cells. It always grows in cluster. It always grows in cluster rather than as single cell. So this growth occurs when the yeast bud do not detach from the parent cell, leading to the visible clusters. So unlike typical yeast, which depends on genetic changes for reproduction, snowflake yeast demonstrate growth mechanism. And this growth occurs when the yeast body do not detach from the parent cell. Growth mechanism of snowflake yeast. The growth of snowflake yeast involves a physical process rather than totally dependent slowly on genetic changes. So researchers observed that these yeast clusters thrive in nutrient-rich solution, grow in nutrient-rich solutions. The solution is highly full of nutrients. They identified two key processes. First one is diffusion and second one is advection. While diffusion allows nutrient movements, it can slowly explain the size of snowflake yeast clusters. And on the other hand, advection, the movement of fluid itself, which plays important role in nutrient distribution within the growing cluster. So in diffusion, it allows nutrient movements. Diffusion allows nutrient to move. So it cannot slowly explain the size of yeast clusters. And in case of advection, there is a movement of fluid itself. So which plays important role in nutrient distribution within the growing yeast cluster. So fluid dynamics are important for the survival and growth of snowflake yeast. As the yeast consume glucose, it differs the density of surrounding solution, which this change creates a flow of nutrients towards the yeast cluster, helping it to grow larger than would be possible through diffusion alone. So the study found that living cluster produce fluid flows that are absent in non-living cluster, which is denoting the importance of metabolic activity in sustaining growth. So there is some implication also. The research challenges traditional views on the evolution of multicellularity. Physical and chemical processes may have initially facilitated and transition from unicellular to multicellular life, independent of genetic changes. So it facilitates the transition from unicellular to multicellular life independent of genetic changes. So this perspective opens new avenue for understanding evolutionary mechanism and origin of complex life forms. So researchers are keen to explore where the principle observed in snowflakes yeast can apply to other evolutionary processes, other evolutionary phenomena as well. So the study raises question about how organism might evolve movement and other complex states based on physical mechanism. The ongoing investigation could reshape our understanding of biology beyond traditional framework, denoting the potential for laboratory findings to inform evolutionary theory. So this is about the current affair till 30th of June 2025. If you like this video beneficial, like, share and subscribe. Hit the bell icon. Do comment in the comment section. Watch the next episode.